Hey everybody, welcome to chapter 2. So in this chapter we're going to talk about microscopy and how we stain bacteria so that we can actually see them under the microscope. So the first thing we're going to do though is we're going to focus on the invention of the microscope. So we're going to kind of skip through the first section, which is a lot of definitions. If I could get my PowerPoint to transition to the next slide, it'd be great. There we go. And we're going right into section 2.2, okay? And so the first individual that we're going to talk about is Antoni van Lindenhook, and he is credited with the first observations of microorganisms, including both protists and bacteria. And in our last chapter, you saw the microscope that he produced. It was a very simple structure. It had a single lens. You can see it right here in this figure from your textbook, B. And this is Antoni van Lindenhook here. He looked at water samples, he looked at teeth scrapings, he looked at semen. So he saw a lot of different materials under the microscope, things that could move. And initially, those things that could move that were super tiny, he called them nanomolecules. We know today that those were either the bacteria or the protists that are regularly found in water or in teeth scrapings. Robert Hooke was one of the first individuals to describe cells. He's actually the one that coined the term cell when he was looking at cork. And when he looked at the cork, he noticed that the cork looked similar, the structures in the cork looked similar to the cells in the monastery. And those were, the cells were where they stayed. That's where their little room was, that they would have their bed and their desk for studying and stuff. And so he coined the term cell. He is one of the individuals credited with the production of the cell theory, stating that all cell, or that the cell is the basic unit of life, and all cells produce new cells. Only living cells can produce new cells. And so this is his microscope. And so these two individuals were given the majority of credit, or are given the majority of credit, for producing microscopes and allowing us to see into the invisible world. But prior to these two individuals, Zecharias Janssen, you can see him over here, is thought to have been the first individual to produce a compound microscope. And so this might have happened in the late 1500s, possibly the early 1600s. But the problem is they didn't publish anything, or he didn't publish anything. He was very secretive. He didn't like anyone knowing what he was doing. So he didn't want to, in science, you know, the whole idea is we want to share our findings so that other individuals can then test what we have done and you know, either agree or find fault in our work so that we can then do more work. That's the idea behind biology and the scientific method. Well, since he didn't publish anything and all of the work that had been done by him has been lost, we can't say that he did invent the microscope. We can't say that he didn't invent the microscope. We can't say anything. We know that he was an individual that did make like simple glasses for individuals so that you could see better. But Leeuwenhoek and Hooke both published, and because they published, we have physical records of them, and so that's why they get the credit, whereas Zecharias does not. So what is a microscope? A microscope is the instrument that allows us to magnify small objects so that we can see them through the lenses. So if you have problems with seeing something, if it's too small, you put it under a microscope. Now you're able to see the structures. You're able to get a better feel for what parts are within that structure. So if you're looking at butterfly wings, you can see the veins in the wings, stuff like that. What magnification does is it enlarges the image. So if you have something that is 10 times magnified, that means it's going to be 10 times larger than it actually is. Light microscopes are used to see microorganisms. So they are one of the most common microscopes used. And using light microscopy, we take visible light, we pass it through multiple lenses, not just one, and then we're able to actually see the microbe at a much larger, as a much larger image. Even when you magnify certain bacteria 1,000 times bigger than what they were or than what they are, you're still going to struggle with seeing the structures. They are very small. Microscopic, haha, right? Resolution definition is the ability to tell two separate points or objects as separate. So here you can see, and this is just an image I found on the internet to show resolution. Here you can see two circles, but they're overlapping. And what resolution does is it separates those two circles. So high resolution allows us to see two individual circles. If it's low resolution, it's going to be fuzzy, and you may not be able to see it as effectively. Oil immersion increases the resolution. So when you're looking through a microscope and you go to the oil immersion lens, you have to add oil. And that lens will sit in the oil, and that oil actually increases the resolution so you can see the images separately. And so here you can see there's the 100 times magnification. This is the oil immersion lens. It allows us to actually, you take this objective lens and you put a drop of oil on the slide, and then this sits in the slide, or the lens itself sits in right on top of the slide in the oil. And with the oil, that light is not going to be refracted very effective, or is not going to be refracted. It's going to go right up, and you're going to be able to see the image. Over here, you see refraction because there's no oil. And so you're not going to see the image. It's going to be very, very blurry if there's any image at all. So different types of light microscopes that are typically used. Bright field microscope, this is what we have in our labs. This is what you'll be using in your lab. We have dark field microscopes. We do not have a dark field microscope, but there are dark field microscopes. And then there are phase contrast microscopes and fluorescence microscopes. Each of these microscopes allows us to see different images a little bit more effectively. So the most common of these is the bright field, and that's why I marked it with red. It's also the one that you're going to be using all the time. So this is a bright field microscope. This is actually one of the microscopes that we have on the Canton campus in the microbiology lab. It looks very much like the microscopes in the Macomb campus. They're very similar. You have ocular lenses up here. Ocular lenses always have 10 times magnification. I would definitely write this down so you remember it. Then you have the revolving nose piece here. The revolving nose piece contains 4 different lenses, or holds 4 different lenses. You have this red line here. This red line is associated with the scanning lens. So scanning lens is 4 times magnified. Then we have this yellow line here. This is with the low power lens, which is 10 times magnified. So you have a scanning and a low power objective lens. Back here you can see a little blue line. That blue line is associated with our high power lens. High power is 10, or is, I'm sorry, not 10, 40 times magnified. So that's a 40 magnified high power objective lens. It is also known as high and dry, which means you have to leave it dry. And so you might wonder, why would I even say that? The reason I say that is because oil is not dry. Oil is wet. And our last objective lens, which you don't see, which will be right over here behind this one, is called the oil immersion, or the oil immersion lens, which is 100 times magnification. So oil immersion lens has to have oil for you to see, but high power over here does not take oil. And actually oil can damage the lens, so it's a dry lens. So you have to make sure when you're using the microscope that you never get oil on the high power lens, but you always have oil for the oil immersion lens, okay? Very important, something that I'm going to stress to you about, because you can ruin the microscopes. These are expensive. This is your stage, and then this structure here, this is the stage clip. So you put your microscope slide in the stage clips, and then right over here we have the stage mover. So this is either going to move the stage forward and back, or right and left. And then we have back here this large knob. This is called the coarse focus. This moves the stage itself up and down. We only use coarse focus on scanning and low power, never on high or oil immersion. And then right here you can see there's the cord here, but you can see the knob right there. That is fine focus. Fine focus is used on all 4 of the objective lenses to tweak the image. There's the light source. You have a button to turn on and off the microscope back here. You have your plug-in right there. So we'll go over all of these also in class, but I want to make sure that you have an idea of what the microscope is and the structures in this video as well. I'm going to stop here, and then I'll pick up on the next slide in my next video, because this has already been 13 minutes. So I will talk to you in a little bit. Bye.